Welcome back to part 2 of chapter 16 on cancer genetics. So previously we were talking about how you can categorize genes that lead to cancer in two different categories. Okay, we got done talking about oncogenes, which are those that tend to turn on something extra that will make the cell more hyperactive and hence enhancing the cell cycle progression. Okay, the second type of genes, identified are called tumor suppressor genes. Tumor suppressor genes are those that causes cell cycle inhibition, okay? And somehow those genes are made inactive when when they have cancer or they lead to cancer, okay? Usually they act in a recessive manner, meaning that we need to turn off both copies in order to stop the function. Okay, kind of like going back to that light bulb idea I was talking about, how you have two light bulbs in a room and you bring in one extra flashlight. That's enough to give you more light, in a sense illustrating what an oncogene is doing. It's just going in there, allowing more things to happen than it's supposed to. Well, for tumor suppressor genes, it's the other way around. You're trying to turn off the light, okay? Turn off the light in the room. Well, there's the room with two lights. You turn off one light. You still have one functioning light bulb, okay, that will give enough light in the room. If you want the room to be completely pitch dark with absolutely no light, you technically have to turn off both lights, okay? Both light bulbs, okay? So that's the same thing for genes. For example, here you have a wild type homologous chromosome carrying the same normal growth limiting factor gene, let's say, okay? If only one of them acquire a mutation that makes that normal growth factor limiting gene no longer functional, that's only turning off one copy. There's that other copy of wild type that can still do its job, okay? And in a sense, if we want to really turn off all inhibition of cell cycle progression, we need to turn off both copies of the gene of the alleles in the homologous chromosomes, okay? That way, both of the genes do not have inhibitory factor, and that can lead to excessive cell proliferation, okay? We talked about this idea of heterozygosity in earlier chapters back from the first half of the semester, and that if you were to lose that one wild type allele, you end up with a disease, okay? This concept was called loss of heterozygosity, where one defective copy of a gene will not result in a disease because the other wild type allele is enough to keep the cell functioning normally. But on the same note as the two-hit hypothesis, all it will take for these to become cancerous is if the other allele also acquire a mutation. You end up losing the heterozygosity that kept the cell functioning normally. So that's what, heterozygous tumor suppressor gene can happen. Okay, you already have one that's not functioning right, and here comes, it's more high. There's a higher chance of acquiring that second mutation that will also make the other chromosome copy completely non-functional. Okay, so although deletion is rare, okay, if we talk about that one deletion that's going to specifically take out that one important gene for regulating cell cycle. It's hard to hit that one gene out of, 6 billion bases you have in your DNA. But remember, all it takes is one cell to acquire the mutation and bypass cell cycle regulation and turn into a clone of cells that starts becoming cancerous, and you will end up with a tumor, okay? So it's hard to hit that target, but it's not saying it's impossible, okay? And especially when you are already heterozygous for a particular mutation, it makes it even easier to acquire just that one more mutation to, have that normal cell turn cancerous, okay? All right. So one of the first tumor suppressor gene to be identified was, of course, retinoblastoma. Okay, retinoblastoma, and that's why that two-hit hypothesis was developed based on the studies of retinoblastoma. Okay, and they named that particular gene retinoblastoma because that gene was found in retinoblastoma patients. Okay, so this became known as the RB gene, which is a tumor suppressor gene, and that you need to turn off both copies of the RB gene in order for a normal cell to become cancerous. Another tumor suppressor that was discovered was, of course, called BRCA1, which was one of those we talked about that functions in DNA repair. When mutation happens on both copies of BRCA1, it will increase the risk of breast and ovarian cancer. And normal gene function of BRCA1 is to help repair double-stranded breaks in the DNA by homologous recombination specifically. So when you have mutation in both copies of the BRCA1 gene, you lose that capability of repairing double-stranded break in that particular DNA repair mechanism. Okay, so that's why if BRCA1 gene mutation runs in your family, a lot of times then you will go and get tested to see whether you also have that particular mutation that tends to run in the family. Maybe you are just heterozygous for that allele. You don't have to be homozygous. You're just heterozygous for that allele. Well, it gives you a probability at least of how likely you're going to get breast cancer. And then the patient will decide whether they want to remove their breast and ovaries to prevent those cells from becoming cancerous. Okay, and a lot of times what people would do is they know they have this predisposition to cancer early on because of family history, and they found that they do have that mutation in their genome. So they would go ahead and have kids, have children, and then after that, then they will go ahead and remove their breasts and ovary and uterus so that, they will completely be clear of having any chance of developing those type of reproductive cancer. Okay, that's what some people will end up doing. And one of the famous case of that is Angelina Jolie, and that's why she went under a double mastectomy, because she found out she has that one BRCA1 gene mutation that makes her a lot more likely to have breast cancer, and it did run in her family. So that's why she had a double mastectomy to remove her breasts. Okay, and that really lowers her chance of having breast cancer. We will talk more about the APC gene, and P53 tumor suppressor gene later in this lecture. Now, contrary to oncogenes, there are a lot less tumor suppressor gene that has been identified, and that's because it's easier to add a bunch of different DNA fragments to a bunch of cells and see which one shows onset of cancer to call it an oncogene. It's kind of like looking, what we talked about with DNA libraries. We're gonna just put all these DNA fragments, clone them into cells, and look to see which one tend to have cancerous phenotypes. Now once we see one group of cells that has cancerous phenotype, then we can further analyze that, whatever DNA fragment we put on that cell, and analyze which gene it is to see what its normal functions are in the cell and what went wrong for it to become cancerous and become an oncogene. Now on the other hand, tumor suppressor genes leads to cancer when it no longer functions correctly. Something turned off. So you end up having to search for that specific mutated gene in specific types of cancer. For example, like I said, to look for oncogene, you put a whole bunch of different genes on different cells to look for cancerous phenotypes. But to look for tumor suppressor genes, you have to find that one or multiple mutated genes out of the entire genome of that cell that led to the cancerous phenotype. It's two different approaches of looking for which genes are responsible. One is a lot easier because you're just putting in something extra and looking for a phenotype. The other one, you're trying to look for that one gene in a haystack that causes this particular cancerous phenotype. So that's why there's a lot less cancer tumor suppressor gene that has been identified. I think the difference is about 90% oncogenes and 10% tumor suppressor genes that has been discovered. Okay. All right, and this image here is what illustrate that loss of heterozygosity or heterozygosity, where even if one was deleted out, you still have that other one that functions normally. Okay. Unless this one also becomes mutated, then you lose that heterozygosity. Okay. All right, so we mentioned that the fundamental hallmark of cancer is, of course, uncontrolled cell division. To find out what went wrong, we have to understand how normal cells can regulate cell cycle, how the proto-oncogenes or tumor suppressor genes control normal cell cycle. Under normal conditions, we know cell cycle progression is tightly regulated, and so cells will only divide when these three things happen. Okay, when additional cells are actually needed, okay, not anytime they want, only when cells are needed, that's when they will trigger cell division. When all components necessary for cell division are present, okay, or when DNA has been replicated without damage, meaning no errors in the genome itself or any of the gene itself, okay? These are usually controlled by products of proto-oncogenes and tumor suppressor genes, okay? So, and those any DNA errors that are present, okay, after cell division, or for example, or before that, usually can be fixed by DNA repair mechanism if the person's DNA repair mechanism was functioning correctly, okay? So some of the key events of cell cycle are controlled by two main groups of proteins, okay? One is called cyclin-dependent kinases, known as CDKs, and the other one is called cyclins, okay? Now, CDKs are enzymes that will phosphorylate other proteins. And sometimes phosphorylation doesn't necessarily mean activation, okay? Depending what protein it is, it can be activation or inactivation, okay? Activation of cell progression or inactivation of things that stop cell progression, okay? CDKs are functional only when they're associated or bound to cyclins. So that's why they're called cyclin-dependent kinases, okay? Kinases means they phosphorylate and they are dependent on binding to cyclin, okay? That's why they named them CDKs. So, how cyclins are regulated in throughout cell cycle. Remember, cell cycles have interphase, which goes from G1, S, and G2, followed by mitosis, which is the 4 phases for going from prophase to telophase, and then finally cytokinesis. Well, cyclin levels actually, or different types of cyclin levels, will oscillate over the course of cell cycle, and they will appear at specific points to regulate progression. For example, this particular cyclin E is heightened during G1S transition, okay? Whereas this cyclin A here is heightened during G2M transition, okay? And there's that cyclin B that happens in the middle of M phase, okay? Maybe that's the metaphase, okay, where cells are supposed to be attached to the kinetochores, and cyclin sort of make sure that happens before progressing into the next cycle. So that's why presence of CDKs and cyclin is what's going to promote the cell or allow the cell to go into the next phase of the cell cycle, okay? Now, because they promote cell cycle progression at appropriate times, cyclin and CDK are known as products of proto-oncogene, okay? So make sure you write that down here. So CDK and cyclin are both examples of proto-oncogenes. because they are promoting something to happen, not stopping something from not happening, okay? They're promoting something to progress. That's why we call them proto-oncogenes. You can remember that as promote and proto, okay? So that's easy way to remember. Oh, proto-oncogene, promote something to happen, okay? Or progress. All right. So only when they become mutated, then proto-oncogenes will turn into oncogenes and promote cell division uncontrollably at a hyperactive mode or at inappropriate times. Okay. The textbook makes the analogy of a car, okay? So proto-oncogenes are like the accelerator in a car, okay? You're supposed to only step on it at appropriate times to allow the car to drive in a controlled manner. You can't, press down on the accelerator pedal all the way to the floor and your car speeds up like crazy, okay? You're supposed to do it in a controlled manner, allowing your car to progress and proceed in a controlled manner. So the mutation of a proto-oncogene to an oncogene is kind of like stepping on the accelerator to the floor continuously, regardless that there are stop signs ahead or even red light ahead, okay? So that's why a car is a good way to distinguish between what proto-oncogenes are doing and what tumor suppressor genes are doing and what oncogenes is like, okay? So proto-oncogenes is that accelerator pedal, okay? You're supposed to press on it controllably, but when it becomes an oncogene, it's like having a haywire accelerator that you keep on pressing it all the time, regardless there's a stop sign ahead of you and you're supposed to slow down, okay? So those stop signs and red light ahead are then analogous to what's called cell cycle checkpoints, okay? These are where the cell is about to divide, okay? They have to pass through all the right criteria before moving on to the next stage. For example, the G1S checkpoint, okay, this is when a cell is just before entering S phase, about to have DNA replication. Usually this is an important checkpoint for any errors present because you're about to replicate all that 6 billion bases of the genome. So if there's a mutation in there, it's going to be replicated. So that's why we need a checkpoint before entering S phase to make sure that there are no mutation to begin with before entering S phase, so we're not replicating something that's going wrong, or we're not replicating an error, okay? And one of those proteins that's involved in this G1S checkpoint is the retinoblastoma protein, or the RB protein, okay? So the RB protein will actually bind to another protein called E2F, okay? And E2F protein is important for stimulating transcription of genes necessary for DNA replication. Remember, we talked about how DNA replications, initiation steps require a lot of transcription factors, okay? So those factors are proteins that need to be transcribed themselves as well, okay? So maybe E2F will stimulate transcription of those genes, okay? Then what RB does is actually it keeps E2F inactive until the correct signal is given by the cell to release E2F, so E2F can then enhance transcription of those genes involved in DNA replication. So this is stopping cell cycle from progressing. So that's why the retinoblastoma gene is a tumor suppressor gene. It stops cell cycle from happening. Think of it, it suppresses a tumor from forming, okay? So it must stop cell cycle at some point until it is the right time to replicate, okay? And here's how it works. During G1, what happens is that cyclin D and cyclin E will continuously increase in concentration and combine with their specific CDK. So you have CDKD associated with CDK, cyclin D, associated with CDK, and cyclin E associated with its particular CDK, okay? See, cyclin D CDK and cyclin E CDK both will phosphorylate your RB protein, okay? And this phosphorylation, once it's all completed by late G1, RB will become inactivated. And once RB is inactivated, it actually releases the E2F protein, and that protein can now act as something that, maybe a enhancer that will activate that promoter region, okay, that will allow for and stimulate transcription of a particular gene that is important for DNA replication, okay. So then once those genes are there, you have all those proteins and enzymes that are made ready for DNA replication, then the cell is able to move on to S phase. That's why without having E2F, these genes will never be transcribed, okay? And hence all those things necessary to start DNA replication won't start, okay? That's why RB protein is holding on to the E2F until these cyclins give it the correct signal and say, hey, we're increasing in an appropriate manner, in the right concentration, and it turns off RB, and then that will release E2F. So that's, in a way, is controlling how cell cycle progresses, okay? So the RB gene product is what keeps a cell from moving on to S phase, okay, uncontrollably by keeping E2F inactive, okay? So that's why RB, like I said, is a tumor suppressor gene, because it keeps a cell from moving on to the next phase. It stops cell cycle progression, okay, until the right signal comes around. Now, when RB is mutated, okay, however, then cells can pass through G1S checkpoint without normal controls, okay? Because RB is mutated, which means E2F is just floating around doing nothing, and it's just going to go straight to transcription of those DNA replication factors. Because, the E2F is no longer bound by RB, okay? So on the same analogy of a car, tumor suppressor genes are like the brake pedal, okay? You need to step on the brake in order to slow down or stop the car from crashing head into a wall, okay? If you know something up ahead will lead to a car crash, you will step on the brake pedal, okay? It's, concept, basic logic. Same way with cell cycle. If they detect presence of a mutation or a DNA error, then tumor suppressor genes will halt or stop the cell cycle to prevent that error from being replicated and become a clone of cancer cells, a different type of crash, So if your brake pedal is broken and you keep stepping on the accelerator to the floor, both things happening at the same time, eventually it will hit a wall and crash, okay? So that's why you can't have brake pedals that's broken. You can't have broken accelerators, just like your cell, okay? For example, if your tumor suppressor genes, which are the brakes, okay, are mutated, your proto-oncogenes have mutated as well into oncogenes, eventually they will become a clone of cancer cells, okay? To add to this story of a car, let's say if your car is already broken, brakes are not working, accelerator just keeps going whenever it wants, you will want to fix it, right? Okay? Well, what if you got a bad mechanic who couldn't fix your car? Okay, this is analogous to mutations in DNA repair mechanism. The cell's tumor suppressor genes are broken, proto-oncogenes have gone bad and start acting like crazy. Now, even the DNA repair system is not functioning correctly. So if mutations are left uncorrected, because your DNA repair mechanism is not working right, cells are multiplying as many times as they want at a very rapid rate and keep replicating those same errors over and over again. This is an illustration of how and why it usually takes multiple mutations to give rise to cancer cells and promote their proliferation in the body, okay? And that's why some cancers can, grow pretty quickly and are very aggressive in infiltrating into your normal cell tissues because they have acquired all these different things that have made them a lot more likely to have this unstoppable amount of cell divisions, okay? So that's why that car story is a very good analogy, okay? Okay, so in the end, here is what I was talking about, how if RB is mutated, that's just like your car not having brakes. If your cyclin D is overexpressed, that's turning a proto-oncogene into an oncogene, that's stepping on your accelerator constantly, okay? So now you have no normal control of cell cycle, and you stimulate cells right through G1S checkpoint like it doesn't exist, okay? So that is something bad, very bad for the cell, and that's how a normal cell can become cancerous, okay? Now, normally when a cell realizes that there are mutations and errors in its DNA sequence, it has already tried to fix and repair the errors, but yet it can't, let's say. Okay, and there are errors that a cell can't repair. Well, there's a last process that the cell can rely on to stop the mutation from being replicated. This last resort we have is called apoptosis. Okay, apoptosis is the process of what's called programmed cell death. Okay, this is where cells' DNA is degraded, the nucleus and the cytoplasm will actually shrink, and all of them will undergo phagocytosis by other cells. So it's just going to be wither away and be eaten by and engulfed and getting rid of by a cell. So that's why it's called programmed cell death. Okay. It's like a last resort for a cell to prevent itself from becoming cancerous. If you ever watch those zombie sci-fi movies, if a character knew he or she would become a zombie, they'd rather kill themselves by jumping off a building, okay? And that's what apoptosis for a cell is kind of like. I don't want to become cancerous, so, I don't want to, cause the death of my host, so I'd rather kill myself, okay, in apoptosis mode, okay, and let normal cells survive, okay? Now, I'm not talking about as if zombie are a real thing or this is happening to a human. I'm talking about little cell, okay? And that's what little cell would do because it knows it has an error that will lead to cancer. The normal mechanism of your body to protect yourself from cancer is to get rid of that cell through apoptosis, okay? However, cancer cells really have found all kinds of ways to make itself viable in our body. One of the tumor suppressor genes essential for triggering apoptosis is called p53 gene. Okay, and not surprisingly, almost 50% of all human cancers express a mutant version of the p53 gene. Okay, and it was found to play a role in many other important aspects of controlling cell cycle. Okay, that's why the p53 gene was the gene that everyone was studying in relation to cancer, because they have found that this was a very important gene that participate not just for triggering apoptosis. Okay, they find that p53 is important for stopping cell cycle. That's what's called cell cycle arrest. Okay, if something goes wrong. It also has to do with recruiting or initiating DNA repair mechanisms. And like what we just said, it does play a role in triggering apoptosis, and also when a cell is able to undergo senescence, which means non-growth stage. That's also involved in p53, okay? And that's why almost half of the cancer out there has this gene mutated, because if this gene is active, technically speaking, there are multiple of things that should be happening correctly to prevent a cell from becoming cancerous, okay? So that's why p53 is a very important tumor suppressor gene. Another thing that our normal cells have to control and limit is the amount of cell division that a cell can occur or have in their lifetime. Okay, this is something that we have already talked about previously in another chapter, because some cells have limited lifetime due to something called telomeres. Okay, remember we talked about telomeres as end sequences of the chromosomes, and however many repeats you have is how old the cell is or how many cell division it has become. Okay, normal somatic cells should only have limited number of cell division. Okay, and that's limited by the shortening of telomeres. Okay. Every cell division, you have a shortening of telomeres. And with more cell division, you're going to get to a point where the telomeres are so short that it's going to trigger senescence, means no growth stage, and also apoptosis, because, it's our way of regeneration of new cells. Okay, once a cell is too old, usually it also could mean that it has acquired some mutation of some sort because it's been exposed to the environment for a certain amount of time and has already went under a couple rounds of DNA replication. Okay, so it is better after some number of cell division to get rid of it and have a new cell, so you can prevent from acquiring too many bad mutation that can lead to cancer. Okay, however, many tumor cells, almost 85 to 90% of them, have a mutated version of telomerase and actually reactivate telomerase in their cells and enable this enzyme expression and acquire the capacity to divide indefinitely and however many times they want. What happens is in cancer cells, you still have telomere shortening after every cell division. What happens is you have telomerase that's been reactivated inside these cancer cells, which adds back the length of the telomere and makes sure the telomere never gets to that shortened point, really short point where it starts triggering senescence and apoptosis. And make sure it never reach that point. Okay, so you always have cancer cells with DNA that has a certain correct amount of telomeres that's going to stop itself from undergoing apoptosis and senescence. Okay, and that's how cancer cells do not have limited lifetime. Okay, they can divide as many times as they want, and you can see the importance of that because almost 90% of cancer cells out there does express telomerase. Okay, so that's why telomerase has been one of the sort of promising target for cancer treatment, because this could be one thing that many cancers have in common, that we can stop its action. Okay, so because it, for sure it's prevalent in 90% of the cell. So if we can stop telomerase from functioning correctly in these 90% of the cell, that means we can kill off 90% of the types of different cancer cells out there. Okay, so that's why anti-telomerase therapy is pretty attractive, okay, in the realm of cancer research. All right, so there are two other abilities that cancer cells have they have to acquire in order to survive and advance to the next stage of malignancy. Okay, remember malignancy meaning they can start cancer cells in other places. Okay, so one is called the process of angiogenesis, okay, which is the growth of new blood vessels for supplying the tumor cells with oxygen and nutrients. Okay. In normal cells, this process is carefully regulated by growth factors normally, okay? And other proteins are also regulating this process, so we don't just end up growing new blood vessels out of nowhere all of a sudden, especially if we are already a grown adult, okay? We won't just all of a sudden start branching off into, more capillaries and arteries and all that stuff, okay? Because we're supposed to have enough already. But in tumor cells, genes encoding those proteins that are responsible for angiogenesis are overexpressed, while inhibitors of angiogenesis are inactivated or underexpressed. So it keeps the inhibition low and turns up those proteins that are responsible for doing angiogenesis, because you have this growing lump of tissue, okay? Well, that growing lump of tissue will become less and less accessible for nutrients and oxygen as it grows really large in size. So that's why they themselves need to trigger blood vessels to form so that it can supply those itty bitty cells in the center of that large mass, okay? Or, to reach every single cell within that large mass of cells, it needs to make new blood vessels. Okay, so that's why the other aspect of attacking cancer, if you look up on the internet, is anti-angiogenesis. Okay, can we stop angiogenesis in a cancer cell, in a sense, starving them to death? Okay. But on the same note, okay, there is a specific type of cancer that is due to mutation in particularly a gene that affects angiogenesis, and that example is called the von Hippel-Lindau disease, or VHL disease. This is where people develop multiple types of tumors, including the brain, spinal cord, eye, kidney, etc. Okay, due to mutation of that particular gene that affects angiogenesis. So this is illustrating that VHL disease and where these tumor cells can form in multiple parts of the body. So that's pretty bad if one has VHL cancer. All right. Okay, so once a cancer cell has acquired enough mutation, some of those will give them the capacity to migrate from its original location to another distant organ in the body. Remember, we talked about this process being called metastasis, okay? So this is a cause of almost 90% of human cancer death, which is what I was talking about in the beginning of the lecture, why when a cancer has progressed to this point in their, stage, okay, this point in stage, it is when cancer is tough to treat, okay? Because now we're not just looking at fighting cancer cells and getting rid of cancer cell in that one location, but we could be talking about the brain, the spinal cord, the colon, okay? These are common places, or even the liver, okay, that cancer cells tend to migrate to through blood vessels, the circulatory system, or the lymphatics, okay, through the lymphatic system. So what they found with genes that has to do with metastasis, the type of genes that are commonly mutated are those that encode components of the extracellular matrix, the cytoskeleton, and adhesion proteins. Remember, that cell need to detach from its original large lump of cancer cells. And usually, like I said, cells tend to like to adhere to each other. That's how we form tissues and organs, okay? But eventually, they will acquire mutations that makes them loosely attach, change their cell morphology, change how their cytoskeleton looks like, and then also change how those adhesion protein works. And now that cell has the capability of breaking away from that large mass of tissues, go into your circulatory system or the lymphatic system, travel to another spot in the body that's distant away from its original site, and start secondary tumors elsewhere. So that's how metastases work. And one of those genes responsible that has been found in pancreatic cancer is called the pallidin gene, okay? This particular pallidin gene is expressed in pancreatic cancer, and its normal gene function is its gene product serves as a scaffold to maintain normal cell shape and movement, okay? Now, if that gene becomes mutated, it leads to a poor cytoskeletal architecture, okay, which means that cell goes from being round maybe to something like this. That's how they usually draw, cancer cells, and ends up looking like if they start changing in morphology. And these cancer cells will become detached from its original location. Either they remove themselves easily, and then they will migrate into blood vessels, lymphatics, and like I was talking about, to other places, and starts forming a lump of tissue somewhere else and cause secondary tumors to form, okay? All right. And this image here actually shows, what capacity that tumor cells need to acquire in order for successful metastasis. And that is why metastasis usually mean an advanced cancer cell progression. Okay, you need somehow, of course, be able to detach, be able to move in circulatory system, okay, have angiogenesis happening, and they're able to adhere to capillaries, adapt to that new environment, survival in circulation. That's another important feature. So all of these things needs to happen within that cell that's able to survive and metastasize to another area. And that's why in order for that cell to have all these mutations that makes them able to have all these things required for metastasis, it usually means it's a later stage, okay? That there's a lot of things that's going haywire within that cell that makes them have all these new capabilities that normal cells don't have, okay? All right, so the last type of genetic abnormality we will discuss in cancer are chromosome mutations. Now, most tumors contain cells with chromosome mutation, okay? And they say that this is both a cause and a result of cancer, okay? Not just cause, or neither is it just result. It's both a cause and a result of cancer, okay? Now, what we mean by that is, let's say we start with some sort of chromosome instability, okay? And chromosome instability can be a translocation somewhere, inversion somewhere, that somehow, it no longer, or it could be something going wrong with chromosome segregation, okay? Now you end up with chromosome at abnormal numbers, okay? That's a form of chromosome instability, okay? Chromosome translocation, chromosome inversion, deletion, duplication, all that stuff is chromosome instability that can happen from sporadic mutations, let's say, okay? Now, once we start with some sort of chromosome instability, that can actually accumulate chromosome mutation, which then can affect individual genes that is located within that particular chromosome, and it may contribute to cancer progression, okay? For example, chromosome deletion can result in a loss of one or more tumor suppressor gene, okay? Let's say you have this normal chromosome and has this one gene that's important to serve as a tumor suppressor gene, okay? Well, if that portion was deleted out of the chromosome, now you have a truncated chromosome. Well, there goes that tumor suppressor gene, no longer there, not active, okay? And chromosome inversions and translocation can actually contribute to cancer in 3 different ways, okay? Now, translocation inversions, either case, you're breaking the chromosome and moving them to somewhere else. So that particular chromosome break can result in disruption of tumor suppressor gene function, okay? Remember how we talked about that there are genes maybe this thick or something within the chromosome? If you cut it at half point, okay, where it disrupts that gene sequence, okay, and then this is moved to another location, or even if you invert it, okay, now you have a chromosome with half of the gene here and half of the gene down here, okay, so that makes it no longer functional, okay, so because you disrupt the gene sequence, okay. Now, the other 2 is pretty bizarre, but it can happen, okay? The other 2 ways that translocation and inversions can cause cancer is that, 1, they can bring together sequences from 2 different genes and generate a fusion protein that happen to be oncogenic, okay? The other last way that translocation can lead to cancer is that potential cancer-causing gene doesn't cause cancer in its original location, but somehow it got moved to a new location where it becomes activated by different regulatory sequences that makes it now an active cancer-causing gene, okay? So there are actually two specific examples in cancer that is a result of these two different types of translocations. Okay, so for example, the chronic myelogenous leukemia, okay, that type of leukemia is specific reciprocal translocation among the long arms of chromosome 22 and chromosome 9. Okay, not the entire thing, but part of it. Okay, so you end up with this part of chromosome 22 switching over with chromosome 9. It's a reciprocal translocation, which means this pink part goes here and this green part goes there. So now this particular chromosome, okay, they call it the Philadelphia chromosome because it was first discovered in Philadelphia. But now you have this CABL gene fused with the BCR gene. Now, normally in their original location, this BCR gene and CABL gene does do its own thing, okay, under normal circumstances. But when these two genes are put together like this, it actually causes a fusion protein that stimulate an increase in unregulated cell division, okay? It's kind of what's the chance of that happening? Two fused proteins doing something they're not supposed to. Well, cancer tells us that this is possible, okay? And this particular cancer is chronic myelogenous leukemia, okay? Something that someone has to undergo chemotherapy for because it's a disease of the blood, okay? Now, the second example that talks about how a cancer-causing gene goes to a new location and becomes activated, and that particular example is Burkitt's lymphoma. Okay, this is cancer of particularly B cells of the immune system. Okay, remember B cells are those immune cells responsible for making antibodies or immunoglobulins. Okay, so they have genes that encode for the immunoglobulins. Okay. Now, Burkitt's lymphoma results from a reciprocal translocation between chromosome 8 and either 2, 14, or 22, okay? Either of those three different chromosomes, okay? Probably because all of these three chromosomes is where you have the immunoglobulin gene, okay? We have multiple places for a different immunoglobulin, okay? So that's why as long as that one chromosome 8, which involves the gene called SMak, okay, as long as that SMak is combined with any of the immunoglobulin gene on these three chromosomes, something crazy happens, okay. This relocates the SMak to be now fused with the immunoglobulin gene, okay? And somehow, because the promoter is around this region for immunoglobulin gene, it actually ends up turning on SMak and activating its cancer-causing capability, okay? It's pretty bizarre, but that's how SMak becomes a cancer-causing gene at this new location, okay? And it will stimulate division of B cells uncontrollably and leads to Burkitt's lymphoma, okay? So both ways, either you're causing a new fusion protein product here in this case, or relocate to a different location that makes that gene behave differently in its original location. Both of those can end up, causing cancer, okay? Okay, so by now we can see how cancer harbors a variety of mutations to make its survival and proliferation a lot more favorable for them. If one was to look at a karyotype of a cancer cell, a lot of times things would look pretty bizarre. And there may be a variety of different chromosome abnormalities, okay? And a lot of these involve extra, missing, or rearranged chromosomes, as we have already talked about. Now, remember, the cancer is initiated when genetic changes causes the genome to become unstable, chromosome instability, for example. It can generate numerous chromosome abnormalities. Once you got one started, it's easier to have more mutations and more abnormal stuff to go on. And if those mutations alter expression of oncogenes or tumor suppressor genes, that's when the onset of cancer will take place, okay? if it happens where the genomic instability does not affect those genes, then we may have something else, or we may have nothing at all, okay? But it has to happen on the oncogene or tumor expressor gene in order to interfere with how cell cycle proceeds in a cell to make a normal cell into a cancerous cell, okay? So mutations in genes that encodes parts of a mitotic spindle apparatus, for example, is a type of genomic instability, okay? It can cause abnormal chromosome segregation and cause chromosome abnormality, okay? And one such gene that became very common cancer gene, okay, is the APC gene, okay, a tumor suppressor gene, okay. So this normal function is for the microtubule, okay. Remember in cell cycle you have microtubule or mitotic spindle correctly forming, and they will attach to the chromosome at the kinetochore, okay. So this attachment becomes very critical for cells to move apart as they should, okay. Now there can be a number of different ways illustrated here that can go wrong in terms of that attachment, and you don't have to know those, but it's just know that APC gene is critical for the correct attachment of the microtubule ends to the kinetochore, so the sister chromatids can be pulled apart correctly, okay. Now, if that was mutated, that particular APC gene is mutated, now you no longer have normal attachment of microtubule to the kinetochore. You can end up with what's called a lagging chromosome somewhere in the middle, and depending where it's closer or where cytokinesis happen, that one extra chromosome can go to one of the cell, and now here's this cell with an aneuploidy with an extra pair of chromosome. Okay, so that's what we talked about too in the previous chapter when we introduced the idea of aneuploidy. Okay, but a particular gene of interest that can lead to that is called the APC gene. Okay. And so, this is an example. Remember, I promised from the beginning of lecture that I will show you a karyotype of a particular cancer cell, okay? So this is a karyotype taken from a colon cancer cell, okay? And you can already see there are a number of things that's going wrong here. First of all, chromosome 1 only has one chromosome, okay? Chromosome 5 only has one. And what is this stuff? Okay, a whole bunch of extra chromosomes that has translocations, inversions. That's why they have different colors on them, because the same pair of homologous chromosome is supposed to have the same color on them based on those fluorescent dye or whatever dyes they use to make this karyotype, okay? So one chromosome having different color means that there's all types of mixing going on with the chromosomes, okay? Chromosome 8 is by itself, and here 9, 10 as well, and then chromosome 11 has 3 copies, okay? Chromosome 12 only has 1 copy, and all these abnormal things going on within the karyotype of this particular cancer cell. So you can imagine how many things can go wrong with with cancer cells. That's why they say, cancer cells is not a single disease, okay? And a lot of times it require tens and hundreds of mutations that can be related to the carcinogenicity of that cell, okay? How likely is that cell to become cancerous, okay? So a lot of number of things is going wrong here. All right. Now, because cancer usually involves multiple mutations, as we now know, okay, a lot of different things can go wrong, and they have overcome many different stuff that your cell already has to keep them in check, okay? So cancer progression usually follows a multi-step process, okay? And hence colorectal cancer is an excellent example of how accumulation of successive genetic defects can lead to cancer, sort of putting the clonal evolution we talked about into a specific cancer example, such as colorectal cancer. So we will follow the steps of progression from an initial benign lump of tissue to becoming a mass of cancer cells. So, first, let's say, one of those cells within the colon, here's your colon, one of those normal cells somehow has undergone its very first genetic mutation by losing a tumor suppressor gene, APC. Okay, so now you have the origin of a benign tumor called adenomatous, adenomatous polyps, So these adenomatous polyps that form in the colon is sort of the initial stage of a precancerous tumor, and we already talked about how inactivation of APC gene actually increases the rate of cell division, Then it acquires a second mutation. Let's say that very lump of cell that, because it's growing so rapidly, acquires a second mutation that leads to activation of an oncogene called RAS. So this give rise to bigger and larger polyps that are now called an adenoma, which is a benign tumor that's actually growing within the colon, remember that what happens with a mutation in the RAS gene, it leads to continuous stimulatory signal for cell division, So this is an oncogene that has gone bad, sort of like the proto-oncogene that has gone bad and turned into an oncogene, So even though growth factors are not there, there's no signal for the cell to undergo cell division, but this mutated RAS gene is just giving the go-ahead signal, go ahead and divide, and it becomes a large lump of cells. Then a third mutation happened. Let's say, very important gene, the tumor suppressor gene P53 is now mutated. Polyp now turns into a type of malignant cell we call carcinoma, So a carcinoma is when cells are starting to change in their morphology, They change in shape and then start to migrate out of its original location. And remember, mutation in P53 gene is going to cause that particular, it will stop apoptosis from happening, and also allow the cell cycle to continue even though there are errors in there, because P53 is required for cell cycle stopping, and also it may be important for recruiting DNA repair mechanisms and factors, So if you have a mutated P53, a lot of those things stop functioning correctly, and now you have a really haywire or chaotic cell, And fourth mutation, let's say the carcinoma now acquires a fourth mutation that leads to other changes or even loss of an anti-metastasis gene, maybe there's particular genes like adhesion proteins that's supposed to keep the cells in place in one spot, but that particular gene has been turned off, or mutated. Now the cells gains this capability of moving away from its original location and making a colony somewhere else, So that will promote metastasis, and you will not only have a lump or mass of cancerous cells within the colon, but those cells have now infiltrated into the lymphatic system or the circulatory system and travel maybe to the brain, to the lungs, to wherever they want to travel to, So that's how, something that began as something very small within the colon can turn into something really bad by the end of its fourth mutation. Now, so that's why we said it is very important to have an early diagnosis, hopefully at this stage of cancer development. So all we have to do is remove those polyps, and you may never have to hit this stage ever again. So, that is why for I think, adults after somewhere in their 50s, I remember you're supposed to do colonoscopy annually, And that colonoscopy is what's going to help you detect presence of these polyps within the intestines and remove them as early as possible, because once it gets to this stage, now you have a very difficult disease to treat, Whereas this, very simple. Remove the polyp and you're done, All right, so we will go through this last slide, which is about how virus can potentially cause cancer as well. Okay, so not very common. Not a lot of cancer is triggered by viruses, but there are particular cancer that is associated with viral infections. Okay, so 95% of women, for example, with cervical cancer are infected with a particular virus called human papillomavirus, or HPV, okay, which you probably have heard of before, okay? And there are also viruses that can cause hepatitis B, and that will actually increase the risk of forming liver cancer. So this is a table that lists the number of viruses, different viruses, and what type of cancer cells they will cause in the host. Okay. Now, many of these viruses that lead to cancer are, again, your retroviruses, or not so favorite type of viruses, as when we talked about types of viruses in earlier chapter. Okay, they can contribute to cancer in two different ways. First, they can mutate and rearrange the host genes themselves, which technically they can possibly convert a proto-oncogene into an oncogene. Okay, so this is that process right here illustrated. So what you have here is a virus infecting this particular cell, okay? Now it inserts its own DNA into the cell, okay? And undergoes reverse transcription, as we remember, okay? It turns its RNA material into DNA material and forms what's called provirus and inserts into the host chromosome. And let's say somehow the provirus was inserted next to a proto-oncogene, so that now your host chromosome has a provirus gene and the proto-oncogene next to each other. And when the virus reproduces and take this particular proto-oncogene into itself, because now the provirus also replicate that proto-oncogene into itself, but what the virus will do to the proto-oncogene, because remember, virus replicates by reverse transcriptase, and that reverse transcriptase itself is very error prone. Okay, and when these viruses go through repeated rounds of infection and reproduction, they can technically introduce mutation into the proto-oncogene itself, okay, and turn the proto-oncogene, which was normal to begin with, into an oncogene. Okay, and what is that illustrating right there with that red dot, okay? That's that mutation the virus has inflicted upon the host, okay? And that mutation has turned a proto-oncogene into an oncogene, and if that particular oncogene now incorporates itself into the host genome, now your host genome has acquired an oncogene, okay? And that can turn on onset of cancer somewhere in the body. Okay, so that's one way. Another way viruses can introduce oncogene into the host is by altering the host expression. Now, retrovirus usually have what's called strong promoters, because they have to ensure transcription of their own viral genome, okay? And that's why they are more likely to be turned on and more likely to be transcribed. Now, if this provirus, again, along with its viral promoter, happens to insert itself next to a proto-oncogene or even close to a proto-oncogene, it will stimulate a very high expression, okay? And that stimulation of the proto-oncogene can also lead to cancer. Remember, proto-oncogenes are supposed to be allowing cell to progress at a control rate. But if it's overstimulated, that's going to cause uncontrolled cell division more than it should, and that can drive your very first mutation to a cancer cell, okay? All right, so this concludes chapter 16 on cancer genetics. Let's go over the practice questions for this chapter, and we will call it a day. Okay, so first question, cancer is a heterogeneous group of disorders resulting from both A and B, okay, rapid uncontrolled cell division, and it is a multi-step process that requires several different mutations, as we have discussed in this whole chapter. It does not accumulate germline mutations during a person's lifetime. Okay, we didn't talk about germline mutations within this entire lecture. Okay. Considering the clonal evolution of tumors, mutations in which kinds of genes would speed up the rate of accumulation of additional mutations? DNA repair genes. Okay, remember we said this is the bad mechanic. So if we have a bad mechanic, this would really speed up the rate of accumulation of additional mutation, because now you have errors that are not repaired by your repair pathways. Okay, so that's what's going to give rise to even more mutations. Which of the following statement is true of tumor suppressor genes? None of the above is true of tumor suppressor genes. Okay, normal function is to promote cell proliferation. Nope, that's for proto-oncogenes. Their mutant forms are typically dominant. Nope, tumor suppressor genes are recessive mutant forms. There were first cancer-causing genes to be identified. Nope, the first one was an oncogene, which was SMak, if you remember. Okay. So that's why none of these describes the tumor suppressor gene. In an experiment, okay, where you mutate the retinoblastoma gene, the RB gene, such that its gene product behaves as if it's hyperphosphorylated, the result would be a blank association between RB and E2F with blank transcription of genes necessary for DNA replication. It will be a weaker association between the RB and E2F, okay, because the gene product behaves as hyperphosphorylated. Remember, we have that RB gene, and we said that these are phosphorylated by CDKs and cyclins. Okay, well, if it was hyperphosphorylated to begin with because of mutation, then that's what's going to release your E2F. Okay. So that's why it's weaker, and it will cause continuous stimulation of transcription of genes necessary for DNA replication, because now you have a continuously released E2F in the cell. The class of proteins known as cyclins oscillates in concentration during cell cycle. It doesn't phosphorylate other cellular protein. What does phosphorylation? CDKs. Not cyclins. Okay, cyclin is just there to bind to CDK. CDK are the ones that's going to phosphorylate other proteins and activate other processes. They don't act as inhibitors of kinases. If anything, they bind and activate kinases. Okay. In normal somatic cells, chromosomes shorten with each generation. In cancer cells, you notice that chromosomes are not shortening. A mutation expression of which gene might lead to this observation? Telomerase. Okay, that's 90% of cancers out there does express telomerase. The APC gene encodes a protein that interacts with the ends of kinetochore microtubules and regulates the rate of cell division. Hypermethylation or decreased expression of the APC gene will most likely result in both B and C, unregulated cell division and increased frequency of chromosome defects. Okay, it's already hypermethylated, down-regulated in expression, so you won't end up with more of those proteins around. Mutations that change chromosome numbers and structure can cause cancer and be the result of cancer. True. Okay, that's what we said. Chromosome abnormality, aneuploidy, can be a cause and a result of cancer. Which of the following is not a way that viruses cause cancer? They do not induce apoptosis of normal non-cancerous cells. Okay, they can cause mutation, rearrangements in the host chromosome. They can convert a proto-oncogene to an oncogene. Their viral promoter can stimulate high levels of proto-oncogene expression. Okay, they can produce a protein that inactivate tumor suppressor protein, but they do not do that. D. Mutations in pallidin gene can cause pancreatic cancer by creating a defect in the cytoskeleton. That's the one we talked about. The pallidin gene resulting in poor architecture of the cytoskeleton that now makes the cell more likely to migrate to other places and start secondary tumors in other areas of the body. Okay, the histone proteins of nucleosomes are often abnormally modified in cancer cells, leading to, now this is a question as a build-up questions from what we learned about histones and nucleosomes, okay? Remember we talked about that in gene expression chapter, okay? So if the histones are modified abnormally, what will happen? There will be alteration in chromatin structure. Remember, histones are important for keeping that nice nucleosome structure, and there will be alteration in levels of transcription. Remember, the acetylation of histones or methylation of histone proteins can lead to transcription or no transcription. So when there's abnormal modified histones, that can actually alter the rate of transcription as well. It is a thought that blank are precursors to formation of colorectal cancer, adenomatous polyps, okay? Adenomatous polyps, which are the benign precancer tumor that can lead to formation of colorectal cancer. The chromosome resulting from reciprocal translocation between chromosomes 9 and 22 is referred to as what kind of chromosome? Philadelphia, okay? This is seen in most people with which cancer? Chronic myelogenous leukemia. Okay, Philadelphia chromosome causes the fusion of the two proteins as responsible for the onset of chronic myelogenous leukemia. Okay, all right, that's all, and I will see you another time. Thank you very much. This wraps up chapter 16.